Structurally, most bacteria consist of a cell membrane surrounded by a cell wall, and for some bacteria, an additional outer layer. Internal to the cell membrane is the cytoplasm, which contains ribosomes, a nuclear region, and in some cases granules and/or vesicles. Depending on the bacterial species, a number of different external structures may be found, such as a capsule, flagella, and pili. In gram-negative bacteria, the gap between the cell membrane and the cell wall is known as the periplasmic space. Most gram-positive bacteria do not possess a periplasmic space, but have only periplasm, where metabolic digestion occurs and new cell peptidoglycan is attached. Peptidoglycan, the most important component of the cell wall, is a polymer made of N-acetylmuramic acid, alternating with N-acetylglucosamine, which are cross-linked by chains of 4 amino acids. The function of the bacterial cell wall is to maintain the characteristic shape of the organism and to prevent the bacterium from bursting when fluid flows into the organism by osmosis. Synthesis of the peptidoglycan and ultimately the bacterial cell wall occurs in a number of stages. One of the first stages is the addition of 5 amino acids to N-acetylmuramic acid. Next, N-acetylglucosamine is added to the N-acetylmuramic acid to form a precursor of peptidoglycan. This peptidoglycan precursor is then transported across the cell membrane to a cell wall acceptor in the periplasm. Once in the periplasm, the peptidoglycan precursors bind to cell wall acceptors and undergo extensive cross-linking. 2 major enzymes are involved in cross-linking: transpeptidase and D-alanine carboxypeptidase. These enzymes are also known as penicillin-binding proteins because of their ability to bind to penicillins and cephalosporins. Eventually, several layers of peptidoglycan are formed, all of which are cross-linked to create the cell wall. Gram-positive bacteria may have more layers than gram-negative bacteria and thus have a much thicker cell wall. Beta-lactam antibiotics include all penicillins and cephalosporins that contain a chemical structure called a beta-lactam ring. This structure is capable of binding to the enzymes that cross-link peptidoglycans. Beta-lactams interfere with cross-linking by binding to transpeptidase and D-alanine carboxypeptidase enzymes, thus preventing bacterial cell wall synthesis. By inhibiting cell wall synthesis, the bacterial cell is damaged. Gram-positive bacteria have a high internal osmotic pressure. Without a normal rigid cell wall, these cells burst when subjected to the low osmotic pressure of their surrounding environment. As well, the antibiotic penicillin-binding protein complex stimulates the release of autolysins that are capable of digesting the existing cell wall. Beta-lactam antibiotics